Sporangium. Microsporangium or the ovule. So microsporangium or ovule, as we know, the sexual part of a plant, that is flower only, right? So flower is the sexual part of a plant. So inside the flower, two reproductive parts are there. The first one is male reproductive part and the second one is female reproductive part, right? So what is the name of male reproductive part? Androecium. Female reproductive part? Gynoecium. Very good, isn't it? So inside that androecium, so in the male reproductive part, that is androecium. So inside the androecium, what is the name of the simplest unit of androecium? Stamens are there, right? Isn't it? So now we can skip that male part of the plant and we can discuss about the female part of the plant. So inside the female part, what is the female part of the flower? Gynoecium. So inside the gynoecium, what is the simple unit of gynoecium? Pistil. Very good, isn't it? Pistil is there or carpel is there. Very good, isn't it? So pistil or the carpel is the simplest unit of gynoecium. Now coming to the parts of the gynoecium. So inside the gynoecium, three parts are there. So can you name the regions of gynoecium? Stamen is in stigma, style and ovary, isn't it? So stigma is there, style is there, ovary is there, isn't it? So inside the ovary, some structures are there. That structures are called ovules. That is inside the ovary, seed-like structures are there. That structures are called ovules.
Actually, ovules are called as megasporangium.
No. Oh, sorry. Okay, so megasporangium, microsporangium, that is another only. Okay, so here that is megasporangium is there. So that is inside the ovary. Inside the ovary, that seed-like structures are there. That seed-like structures are called ovules, or else we can call it as megasporangium, isn't it? So coming to the structure of this megasporangium. So about the structure of megasporangium or the ovule. So inside the ovule, the first region, that is the stalk-like structure. The stalk-like structure, that is called funicle. So you know that is inside the flower, ovules are there and ovaries are there. So once after the fertilization, so fertilization means after the fusion of male and female gametes. So once after the fertilization, the ovary is converted to fruit and the ovule is converted to seed, isn't it? So ovule converted to seed and the ovary is converted to fruits also. So here the seeds having one stalk-like attachment. The seeds are attached to the fruit, right? So that attachment portion, that is called funicle. That is the stalk of ovule or the seed. Stalk of ovule, that is known as funicle, and the attaching portion of this ovule, or else we can call it a seed. After fertilization, this seed or the ovule that attaches with the fruit or the pod, that structure is known as hilum. That is the attaching portion of seed, attaching portion of ovule or the seed with the fruit, that is known as hilum. So once again, I'm repeating once again. The first one, that is about the stalk of ovule. So stalk of ovule is known as funicle, and the funicle that attaches with the ovule or the seed, that is called hilum. So clear? Okay. So when you're observing a seed, suppose if you are taking a pea seed, isn't it? Seed of a pea plant. So inside that one, the upper portion, so maybe like this one. So in this side, like scar-like structure is there, or this is the opening region. That is the opening region in which plants are germinating. So while the saplings are germinating, the seeds are the plants or saplings are coming out from that particular area. That is known as the opening region. That opening region of the ovule, that is known as micropylar region. So micropylar regions are there. So micropylar region means here, that is, it is the opening region of ovule. So opening region of ovule, that is known as micropylar region. So once again, inside the seed or the ovule, opening regions are there. That opening regions are called micropylar region. So micropylar region through which the saplings are coming out, isn't it? So just opposite to this micropylar region, the opposite region that is known as callosal region. So writing is different and pronunciation is different. That is callosal. Okay. So callosal region. So inside the callosal region, so here that is the region opposite to micropylar region. So the region that opposite to micropylar region, that is called callosal region. Okay. So here some specific cells are there. Later we can discuss after completing the structure of ovule. After that we can discuss about the differences in the micropylar and ovule region. Okay, or callosal region. So once again, opening region is there. So opening region is known as micropylar region, and opposite the micropylar region, that is the next to that one, that is callosal region. That is the region opposite to micropylar. Is it clear? Any doubts in this area? Anyone? Fine. Next one, this ovule is surrounded by one protective layer, and this protective group of layer is called integument layers. Very good. That is known as integument layers. So integument layers are there. That is the region that surrounding the ovule or the protective covering of that ovule, that is known as callosal region. Sorry, integument layers, isn't it? That is known as integument layers. So here, this integument layers further classified into two. That is outer integuments are there. That is the outer portion. The first layer, that is outer, and the next one, that is inner integument also there. The inner region, that is inner integuments are there. So once again, integument layers, that is the protective layers of that ovule, that is known as integument layers, and this integument layers further classified into two types or two layers. That is outer integument layers are there, and the next one, that is inner integument layers are there. Okay, so two types of layers are there. So its function, that is the protection, that it protects the whole structure, or else we can say that it is the protective layer of the embryo or the embryo sac. Now here, inside to this one sac-like structure at the center, and this sac-like structure, that is known as embryo sac. So embryo sac is there, that is a sac-like structure that present inside the ovule, that is called embryo sac, or else we can call it as female gametophyte. It having one more name, that is known as female gametophyte. Female gametophyte, that means there is a gamete producing structure inside the plant body. The female gamete producing structure inside the plant body, that is called female gametophyte. So clear? So once again, at the central side of the ovule, embryo sac is there. One sac-like structure is there. The sac-like structure we can call it as embryo sac. So that embryo sac that helps in the production of female gametes. Is it clear? Okay, there is embryo sac is there. Now, outer to this embryo sac is covered by one nutritive layer, and this nutritive layer we can call it as nucellus. So remember, nucleus also there, nucellus also there, both are different, isn't it? So what is nucleus? Controlling part of the cell, isn't it? So controlling center of the cell, that is called nucleus, that is different, and here nucellus is there. So nucellus, that is the nutritive layer that surrounds the embryo sac, that is called nucellus. So clear? It provides nutrition for the developing embryo that present inside the embryo sac. Okay, so instead of explaining this structure here, we can explain it separately. That is the central structure, that is sac-like structure. So about the structure of embryo sac. Okay. About the structure of embryo sac, first of all, remember, can you count the number of cells that present inside this cell, embryo sac? 7 cells and 8 nucleated. Very good. So okay, so it is formed from 8 cells, but 2 cells were fused together to form a single cell and becomes 7-cell structure, right? Okay, so remember, embryo sac is a 7-celled 8 nucleated structure. It having 7 cells but 8 nucleus. That is 7-celled 8 nucleated structure. Okay, so one by one, first of all, how to identify So here in the ovule, central side embryo sac is there. So inside the embryo sac, remember one side is micropyla and opposite region is callosal region. So how to identify the micropylar and callosal region? So remember the region that having different type of cells, that is micropylar. Okay, so if you are go through different types of materials, it will be reversed. Sometimes micropylar at the lower side, sometimes micropylar at the higher side. They will represent like that one, isn't it? So how to identify means the area that having three different cells, that is micropylar, and the area having three similar cell, that is callosal cell. Okay, so here this region we can call it as micropylar region. So remember, in the micropylar region, three different types of cells are there. Okay, three different cells are there. And opposite to this one, that is callosal region. Callosal region, isn't it? So it's written as callosal, but read as callosal. So callosal region, that is opposite to the micropylar region. Okay. So remember here, in the callosal region, three cells are there, and these three cells are known as antipodal cells, and they are similar cells too. So these are antipodal cells, that is similar cells. The three cells present at the callosal region, those cells are similar in nature, isn't it? So we can call it as antipodals. So what about the ploidity of the cells? Is it haploid or diploid? Ploidity in the sense about the number of chromosomes. As we know, what about the number of chromosomes in human body?
12 in somatic cells and 1 in cells.
Can you stand up, please?
Diploid in somatic cells and 1 in sexual cells.
Okay, so diploid in somatic. Okay, so what about the number of that chromosomes?
23 pairs.
23 pairs of 46 chromosomes. Very good. So, okay. So in human body, total number of chromosomes, that is 46 chromosomes are there. That is in diploid in condition, or else, isn't it? So diploid in condition, that is 2n number of chromosomes are there. Suppose if you are observing the gamete cells. So in the case of gametes, the diploid chromosome number converted to haploid, right? So that is 23 pairs becomes 23, or 46 chromosome, it becomes 23 number of chromosomes only. So this 46 chromosomes converted to 23 numbers by a process. What is that process? Meiotic division, isn't it? So as a result of meiosis, diploid cells convert into haploid cells. So here also like the same, the formation one process is there, I think already discussed in classroom before, right? So that is, have you learned megasporogenesis? Or else later we can discuss. Okay, so here also like the same, that is the embryo sac is formed as a result of meiosis. So meiotic, after the meiotic division only, embryo sac formation takes place. So here inside the embryo sac, this antipodal cells also having the chromosome, that is n number of chromosomes only. Okay, so based on the plant it will be differs. Chromosome number is different types of plants. Based on the plant, again chromosome number differs. So here that is antipodal cells are there. So antipodal cells are the three similar cells that present at the callosal region. Is it clear? Boys, clear? Fine. Okay. Now opposite the callosal region, in the micropylar region, this whole structure we can, or the three cells, the group of three cells we can call it as egg apparatus. So egg apparatus is there, that is different cells present at the micropylar region, we can call it as egg apparatus. Clear? Okay. So inside the egg apparatus, one large cell is there, and this large cell we can call it as egg cell. We can call it as egg cell, and two cells are there, it is almost similar, we can call it as synergids. So once again, inside the egg apparatus, three cells are there. So one large single cell is there, that cell we can call it as egg cell. So what is the function of egg cell? What is the use of that egg cell?
Later it fuses with the male gametes and develops into seed.
Okay, so stand up. Repeat it once again.
Later it fuses with the male gamete
Ah.
and develops as a seed.
Okay.
Another male gamete it fuses with the central nuclei and it becomes as primary endosperm.
Okay, fine. Okay, that's about the whole process. Fine. Okay, here that is egg cell is there. So egg cell that helps in fertilization. So fertilization, that is the fusion, isn't it? So fusion with the male gametes. So once again, egg cell is there. So in the egg apparatus, egg cell is there, that is a large cell, and also syringe is also present. Now the next one, here certain projections are there, small projections are there near to the egg apparatus, that is called filiform apparatus. So here some projections are there that we can call it as filiform apparatus. This filiform apparatus that helps to try the male gametes, isn't it? So during fertilization, so the male gametes are there. When the male gametes are transferring from the upper region, so that time it pushes the male gametes towards the egg cell. That is a function of filiform apparatus. So once again, filiform apparatus is there. So they are the finger-like projections that helps in fertilization by pushing the male gametes. Clear? Now, at the central side, two cells are there. So two cells join together to form or fuses together to form secondary nucleus. The central structure we can call it as secondary nucleus, or else polar nuclei. Very good. So we can call it as polar nucleus. So once again, at the central side of embryo sac, two cells fuses together to form a single nucleus, that is called secondary nucleus, or else we can call it as polar nucleus. So clear? So once again, about the structure of embryo sac or the female gametophyte. So in the embryo sac structure, the first region, that is micropylar region. So micropylar region having three cells, that is egg apparatus. In egg apparatus, single large cell, that is egg cell is there, and two synergids also there. So along with that one, filiform apparatus is there. So filiform apparatus that helps to pushes the male gamete towards the egg cell. Now the next one, that is callosal region. In the callosal region, three similar cells are there. We can call it as antipodals. And the next one, that is secondary nucleus, that formed as a result of the fusion of two cells from one from micropylar and one from callosal region. Okay, so that is the secondary nucleus. Now inside this embryo sac, two processes there, that is called double fertilization. Already she mentioned, that is double fertilization and also triple fusion. So first one, that is double fertilization. So double fertilization, before that one, have you learned the structure of microspore? Right. So what is microspore? Can you give the common name for microspore? Pollen grain, isn't it? So pollen grain, that is the microspore. So inside that pollen grain, how many layers are there? Two layers are there. Outer layer is exine, inner layer is intern. So two outer layers are there. How many cells are there in pollen grains? How many cells are there? Two cells are there. Generative cell and vegetative cell, isn't it? So generative cell is there and vegetative cell is there. So which one is the largest? Vegetative. Smallest one is generative, isn't it? So generative is there. So generative cell can able to produce male gametes, right? So inside the microspore, generative cells produce male gametes, isn't it? So from a single generative cell, how many male gametes are producing? Two male gametes are producing, isn't it? From a single microspore, two male gametes are producing. So that two male gametes are there. So one male gamete fuses with the secondary nucleus and one male gamete fuses with the egg cell. So from a single microspore or from a single pollen grain, that is two male gametes are there. So one male gamete fuses with the secondary nucleus and the second male gamete fuses with the egg cell, isn't it? So here, the first one that fuses with the egg cell and results in the formation of zygote. So here, that is male gamete is there. So one male gamete fuses with the egg cell and results in the formation of zygote. So what is this process? Fertilization, isn't it? It having one more name. What is that? Syngamy, isn't it? So fertilization and syngamy both are same only. So here the fusion of male and female gamete, that is called fertilization, or else we can call it as syngamy. Next one, the second male gamete that fuses with the secondary nucleus and results in the formation of a new structure, and this structure is known as primary endosperm nucleus. primary endosperm nucleus, or else we can, in short, we can call it as PEN, or else one more name. What is that? Simple term, familiar term only, endosperm. Okay, so endosperm. What is endosperm? Nutritive tissue, isn't it? So nutritive tissue inside the embryo sac, that is called endosperm. So remember, the polar nucleus or the secondary nucleus, that is formed as a result of the fusion of two cells, one from the micropylar region, and one from the callosal region, isn't it? So two cells fused together to form secondary nucleus. Along with the secondary nucleus, one more cell is fusing, that is male gamete is also fusing, male nucleus is also fusing. So here the fusion of three nucleus takes place, so that we can call it as triple fusion. Okay, so remember, triple fusion is there. Triple fusion means the formation of endosperm, or else the formation of primary endosperm nucleus as a result of the fusion of three nucleus. Hence we can call it as triple fusion. So triple fusion means that is the formation of primary endosperm nucleus, or else endosperm. The formation as a result of the fusion of three nucleus, that is called triple fusion. Is it clear? So these two processes together we can call it as double fertilization. Very good. Together we can call it as double fertilization. That is the fertilization that takes place twice inside the embryo sac, that we can call it as double fertilization. So the first process is syngamy and the second process is triple fusion. Both syngamy and triple fusion together we can call it as double fertilization. Is it clear? Okay. Any doubts in this area? Anyone?
Sir, may I know the difference between the rules of nucellus and endosperm?
Endosperm, isn't it?
Differences between the rules of nucellus and endosperm.
Okay. So, any other? Okay. So nucellus that present inside the ovules only, isn't it? So remember, nucellus is outside this embryo sac region, and next one, that is endosperm, that present inside the, which one? Present inside the embryo sac only, isn't it? And inside the embryo sac only. Now about the differences, the first one, that nucellus is a nutritive layer. Already we mentioned the nucellus is a nutritive layer that surrounds the embryo sac, that helps in the embryo sac formation. Main function that helps in the embryo sac formation, in the sense, one process is there, I think already discussed in classroom. In short, I will explain. Okay, that is megasporogenesis. So in the megasporogenesis, as we know, the embryo sac is there. That is 7-celled 8-nucleated structure that is formed as a result of meiotic division and mitotic division. It starts from a single cell. A group of tissues there, that is sporogenous tissue. So from the sporogenous tissue, one of the cell acts as megaspore mother cell, and this megaspore mother cell absorbs nutrition from the outside. Okay, then megaspore mother cell undergoes meiotic division, again 2-cell stage, 4-cell stage, and finally 8-cell stage. It's a detailed process, later we can explain. Okay, that is megasporogenesis in short. So during the megasporogenesis, this megaspore mother cell absorbs nutrition from the surrounding cells, that is from nucellus. And next to that one, during the fertilization, during the double fertilization, egg cell is converted to zygote. Later this zygote develops into embryo. So that process is called embryogenesis. So when zygote is transformed into embryo, that time also a lot of divisions are there. I think in zoology in detail you learned about that concept. That is, first one, zygote is a 2-cell stage, isn't it? Male gamete and female gamete combine together to form zygote. Zygote undergoes division to form morula, basula, gastrula. Different stages are there. 2, 4, 8, 16, 32, 64. Different stages are there. So during this division process, they also require some amount of nutrition. So like the same here also, during that division, particular division or the formation of embryo, this zygote or else this embryo is absorbing nutrients from the inner portion. Is it clear? So both are the different cases. Is it clear? Okay. Now, any other doubts if you have, you can ask.
Anticoagulant cells and synergids are formed by meiosis. And why they are different in size? Like anticoagulant cells are similar and synergids, the agapparatus is different.
Agapparatus is different, isn't it? So remember, first of all, during the fertilization, isn't it? So during the fertilization, only one cell can able to act based on the formation only. So there is a modification. So out of all the cells, some cells are modifying. So this egg apparatus is modifying for the fertilization. That's why it is different. For example, compared to the human body, isn't it? So the human case also, ovum, the releasing of ovum, lot of sperms are combining with the or fusing with the ovum, but only one sperm have the ability to fuse with the ovum, isn't it? Already you learned in second chapter or the reproduction chapter, isn't it? So like the same here also, all of the cells that present inside the embryo sac can be able to undergo fertilization. So some of the cells are modifying. That modified cells are in egg apparatus that is fusing with the male gametes. Clear? Fine. Okay. Now a few points along with this one. Some other additional points are there. So antipodal cells are haploid in nature, right? Okay. So what about egg apparatus? In haploid only, isn't it? So each cells in the egg apparatus is also haploid in nature. If you are taking synergids also, or else egg cell also, all these cells are haploid in nature. So remember, one of the most important question that is in for the NEET also, and for about the ploidity, you can expect in match the following type of questions, isn't it? Okay. So here the next one, that is central cell is there, polar nucleus is there. So in polar nucleus, what about the number of nucleus inside the polar cell? 2n. So remember, polar nucleus, the ploidity of polar nucleus, that is 2n in condition. And what about endosperm or the central cell? 3n condition. Why? Triple fusion, isn't it? The fusion of 3 nucleus, so 3n, that is the condition. Clear? I think ploidity is over. Okay, fine. So any other doubts?
Will the new cells disappear after the formation of embryo sac?
New cells, isn't it? So after the formation of embryo sac. Okay. So both conditions are there. Sometimes they can able to absorb the whole nucellus also. Endosperm also same. Okay. So during the germination or the development, sometimes they will absorb the whole thing, or else remnants of nucellus is there. It's already mentioned in NCERT also. It's already mentioned. Remnants of nucellus remains after the fertilization or the development of embryo. That also possible. Not in all the cases, isn't it? For example, in the case of coconut, that liquid, isn't it, or the watery substance, that is the endosperm. So it is not consumed during the development. So the remnants of that endosperm is there inside. So like that, nucellus also present. So clear?
But there's a time gap between the fertilization and the development of embryo sac.
Fertilization and
Embryo sac, formation of embryo sac. And within that time period, what is the role of new cells?
Role of nucellus. So that time, so first of all, after the division, so they need that is called dormancy period. Suppose if you are taking a simple seed, for germinating, it needs some amount of time. It needs some amount of time to germinate, right? So the waiting period, that we can call it as waiting period or gestation period or dormancy period. The dormancy period, what they can do means the cells or the cells that present inside that particular one, that can able to, that becomes what you can say, adaptive, isn't it? It becomes adapted to the environmental condition, and the cells will proliferate and they can able to absorb. So that time they are absorbing the nutrients and adapting with the environment. That is only process takes place during the dormancy period. Okay. Okay.
I just have few questions.
Okay, ma'am.
I know, for example, in this NEET examination, there is a question stating that what's the purpose of the micropyle? Okay. Instead of we can say that micropyle region, it's generally called as micropyle. Okay, at the size. And now micropyle, do all the pollen grains enter through the micropyle?
No.
The other option is pollen tube. Which one enters through the micropyle? Either the pollen grains or the pollen tube.
So concept is different types are there. So about the structure of ovule, lot of different types of ovules are there. Like anatopous ovules are there. Like different types of ovules are there, and in each and every ovule, the entry of pollen grains through different areas. Sometimes it will enter through the micropylar region, and sometimes it enters through the callosal region, and sometimes it through the integral layers are there. So based on the entry of pollen grains, different types of ovules are there.
Okay, now the question is, there may be 4 options.
Hmm.
Okay,
I don't know.
out of 4 options, one is pollen grain.
Ah,
Okay, the purpose of micropyle is one is pollen grain, entry of pollen grain or pollen tube.
Okay.
Which option should I choose?
pollen grains. Pollen grains are entering inside. So filiform apparatus, with the help of the filiform apparatus, it pushes the male gamete inside.
Okay, and what happens to the integuments? The
Integ-
outer and the inner integument.
Later it will be disappeared. So during the germination, it will be disappeared later as a result of fertilization.
Now, how do seed pods are formed then? Seed pod. The ovary becomes the
Ovary becomes
ovary converts into
fruit,
fruit and ovary converts into
seeds
seed. And now, how do the seed pods are formed?
from outer layers only. Outer layer.
Huh?
From outer layers. Oh,
I couldn't get you.
from the outer layers only, ma'am. That is integument formation.
Integuments, yeah. Okay, integuments convert themselves into the seed
Seed
pods.
pod.
Okay. And what is the purpose of the synergids?
Syner-
Function of the
synergids
synergids.
that in the fertilization process they are supporting the egg cell.
They are supporting
I think so.
Okay, which ovule is very much common in the plant kingdom? There are
Anatopus.
too many types of ovules.
Anatopus.
Okay. Anatopus.
Okay, thank you.